So I'm going to talk about CRISPR-mediated technologies and technologies that are already moving beyond CRISPR in various interesting ways. First, I want to say thank you all the way through the talk. I'm going to give thanks, but in particular to the first and middle authors of papers that we've written together. And these include, these are covered in somewhat by Heidi Leffer's paper, but all the way back to Bernard Dujon, with whom I worked with on in the 1970s, and on zinc fingers and talons, as well as CRISPR. CRISPR was a discovery. It was biochemistry until 2013, when it became a technology, and a technology in human cells in particular is the first eukaryotic cells. And this required at least 8 different things to happen that were not merely finding something that's present in nature. And this included not going beyond just cutting and beyond non-homologous end joining, or NHEJ, to homologous recombination, to editing in normal cells, not just cancer cells, normal cells being a little bit harder, to avoid off-target. We used a computer search. We had to have the right guide RNA that was long in 1 unit, so-called single guide, and we had to get the right expression for that guide RNA, and we had to make libraries and multiplexing. And I'll develop some of these themes in just a moment. And we had lovely company from Jennifer Doudna, Feng Zhang, and their coworkers. So what's better? What would be better than CRISPR? I don't think CRISPR needs any introduction, so I'm jumping to what's better, which is sometimes genome reading alone will do it, and I'll show you an example at the end. Classical gene therapies are just adding genes, not necessarily using a cutting enzyme. Deaminases change one atom, nitrogen to an oxygen, C to T, changing a base pair CG to AT, and or A to G. And then finally, recombinases, integrases, and transposinases allow us to put in bigger pieces of DNA and highly efficiently. There are a variety of ways of getting this into the clinic, into therapies. These include making a mess, which we call in the genome called end joining, precise editing, base editing, and most recently gene writing, where you make these big changes. And Tessera is an example of a company that's doing that. Now, just to illustrate the importance of multiplexing, being able to multiple edits, either together or spread out throughout the genome, it will illustrate this with making organisms resistant to viruses. For example, you can make humans resistant to HIV with 1 edit in the CCR5 gene. We made pigs, we completely eliminated the endogenous retroviruses from pigs with 25 to 62 edits. And we made an industrial organism resistant to nearly all viruses, not just HIV or endogenous retroviruses, by 321 changes. And the reason we've been able to ramp up the number of edits is also related to how well we can read DNA. Reading and writing DNA have both gone up very quickly, exponentially, 60 million-fold improvement in quality and cost. And so, for example, reading DNA has gone from the $3 billion base pair genome to the $300 6 billion base pair diploid genome, which is of high quality and clinically useful. And this has mostly been due to multiplexing, meaning being able to mix samples where you can mix hundreds to trillions of samples in a single droplet and treat it as if it were a single reaction, same cost. Now, for extensive precise editing, we have these recombinases and integrases. Examples are lambda red system and some of these phage integrases, as well as human examples as well. And here, some recombination occurs without making double-strand breaks, and so it's less toxic and less challenging to the cell to repair. Or we could work at the replication fork. And an example where we use both methods, both the integrases and the recombinases, to engineer 321 changes genome-wide. All these little blue dashes are placed in the genome that we edited. We achieved 3 goals, which was new amino acid chemistry, biocontainment, and multivirus resistance. We think that by this method we can make any organism resistant to all viruses in a way that's escape-proof. I know that's a bold claim, but I'll show you data in a moment that supports that. And this was work to wonderful teams led by Mark LaJoy, Dan Mendel, and Nelly Ostrov, who's still leading the team in our group. So here's data from Farron Isaac's team. Farron was part of the original team in our lab. And in this case, showing all these viruses on the bottom line here, 5 of them out of 7 had complete resistance to viruses. We could put in titers as high as 1 trillion viruses per mil, plaque-forming units per mil, and we could recover 0 viruses. This is from one strategy. It's 321 changes, but it's one strategy. These are edits, and these edits were made without CRISPR. They were done with recombinases and integrases. Another example of multiplexing, but one that's already in the clinic, is knocking out various genes that are helpful for the CAR T therapy of cancer, typically B-cell cancers. The CAR T targets the B-cell, and then you have somewhere between 1 and 5 different mutations in things like the T-cell receptor, the major histocompatibility, PD-1 pathway, and so forth. And so these edits, just knocking them out is easy for nucleases like Talon's CRISPR zinc finger nucleases to eliminate, to make a mess of a gene. And that was its first clinical success. And here's one of the first babies that was saved from a very early age B-cell leukemia or lymphoma. Jump from there, where we're making 1 to 5 edits, to now we have 42 edits made in pigs in the wish list of the entire field that's accumulated over about the last 20 years, affecting sugars and clotting and major histocompatibility, other immune functions, and so forth, including all of the endogenous retroviruses. For the first time, we have a pig that doesn't make endogenous retroviruses, which are potentially a threat that the FDA was not prepared to accept, the threat of going from the pig cells to human cells, where we know they will replicate and infect additional human cells. So we did not want to have a zoonotic disease evolving in an immune-compromised transplant patient. So this is for transplants. We can make enhanced versions of it. We have 42 gene changes. We have about 2,000 pigs in the world that have been engineered in this sort of way. Most of them have not gotten all 42 changes just yet. And all of this work is done at Keihan in Hangzhou and eGenesis in Cambridge, Massachusetts. Jim Markman has led the surgical team at Massachusetts General Hospital that have tested these organs in non-human primates and have excellent survival on the order of 300 days. And then hopefully this will be transitioning to human clinical trials soon. Now these were done, quite a few of these were done with CRISPR. Most of these edits were done with CRISPR, but some of them were done by a knock-in strategy that did not, that was more complicated than that. Now that has some toxicity. In order to achieve this, we had essentially 0 cells surviving 25 hits at once, 25 CRISPR hits at once. So we used 2 factors, a fibroblast growth factor and a p53 inhibitor, and now we got most of the cells were surviving and producing the kind of clones that we would take the nucleus out, put it in a pig embryo, and get next generation of pigs. But a far less toxic approach was developed both by Luhan Yang in our group and a number of people in David Liu's lab. They were really, David's quite pioneering in this, changing C's to T's and A's to G's. And I'm not going to go through that other than to say that it does it without introducing double-strand breaks. It can, especially the C to T, is not quite as good as the A to G for 2 reasons. One is it can introduce nicks during a repair process, and second, it has more off-target, at least so far. But all these things are subject to change as kind of the theme of this talk is we're going beyond the previous editing methods. So I already mentioned endogenous retroviruses. These are modest numbers of 25 to 62 changes in the genome at once. Still a record for CRISPR editing. We've gone on to use these deaminases, so getting away from the double-strand breaks of CRISPR, to edit a much larger family of line elements. These are long interspersed repeats that are found in the genomes of many mammals. And in particular, we're looking at the human lines. And these are not just because it's an interesting challenge, it's because these repetitive elements are involved in senescence, neurogenesis, cancer, and inflammation. And we would know more about them if we could edit them. Editing sequences, many of these sequences are hard to sequence, and they're hard to read, and they're hard to write, but we have these new tools in the deaminases. So we use these deaminases, CRISPR-connected deaminases, to edit either C's or A's. And we had to be very cautious to avoid all sources of nicking, including repair processes like mismatch repair and uracil glycosidase to repair the C's to U's and then to T's. And then we had to use the same protective molecules we used that I mentioned before for the pigs. This is work for Corey, Oscar, Khalid, and Verena. And the preliminary aspects of this has been published now. But unpublished and a much bigger record here is we're looking at human stem cells. So we started with cancer cells, which are about 3 or 4 times easier. Our record now for engineering human stem cells is almost 23,000 edits simultaneously in one cell. And this is done by putting in a guide RNA directed at the line element and directing A to G transitions via this deaminase. And this is Raphael, Corey, and Oscar that did this lovely work, which will be published soon, hopefully. Another example where we want to make a large number of changes, not necessarily edits. This is done by adding genes, not CRISPR. So in a way, this is beyond CRISPR. It also predates CRISPR. This is kind of classical gene therapy with a non-classical goal. However, the goal here is that aiming at most diseases that have an aging component. A huge fraction of morbidity and mortality in humans is related to, is exacerbated by aging. And so we picked 5 different diseases. These were reported in PNAS and Protein Cell and some related work in Nature. And we could tackle all 5 of these diseases with 3 genes so far. This gene list will probably grow and be modulated in various ways, but it was striking that we could get significant results on all 5 diseases with these 3 genes or subsets. This is work from Noah Davidson, who now started the company Rejuvenate Bio in San Diego, to extend this to dogs and to humans. This was originally done in mice. So, epigenomics. So that last application was done using adding genes, ideally systemically to every cell, as many cells as possible, but protected against the non-delivery to all cells by aiming at non-cell autonomous, meaning that whatever cell did get the DNA, the added gene, would then spread that added gene via the blood to every cell in the body. Now with epigenomics, what we're doing, rather than adding a gene or editing a gene, we're changing the expression of a gene. Now we might change the expression of a number of genes by adding one gene, but the goal is not so much to add that gene because it can be extinguished once you get the system in the right place. The idea is to change the epigenetic state, the way it's regulated. Now this can be done with microRNAs, with splicing factors. We chose transcription factors. These are very powerful and very general, as you'll see in just a moment. This was done by Alex Ng and Peristu Kozlachag. And these, we have a library that we think has all of the DNA binding proteins in the human genome in expression vectors that can be popped by a gateway into a variety of other vectors, either AAV, lentivirus, or piggyback. We like piggyback the best because it has the largest payload, and it has the highest efficiency, highest expression level. So we've done this with many different cell types. We now have recipes for about 290 different cell types, and that is growing very quickly. And we use as a starting point the transcriptome of the cell, of the target cells, ideally a single cell sequencing of the RNAs to quantitate which ones are expressed at high levels. And then we basically pick the transcription factors from that would achieve that goal. And then we make a number of different combinations. Sometimes we can do it with one transcription factor. Usually we have to use combinations of transcription factors, which requires a combinatorial search, which is not possible to do completely exhaustively, but we can use the transcriptome as a guide. So using these 1,700 putative transcription factors in expression form, we've got 290 cell type recipes, and here's three of the simplest ones where they're single factors, not combinations, that happen to work to make endothelial cells for ETB2, fibroblasts, and ATOH1 for neurons. And you can see the transcription heat map here for these three cell types with many different kinds of transcription factors along the x-axis. So we have many recipes for each of these cell types, so it's showing there's multiple ways to get to a very similar place in the system, not necessarily exactly the same place. This is unpublished. It should be in press any day now in Nature Biotech. And we can make combinations of cells. So we can make pure cell types, some with 1 to 7 transcription factors. We can make combination of cell types where each stem cell is programmed with a different cocktail of transcription factors. And then these can be merged together in a mixture of cells where the ratio of the different cell types is determined in advance before they differentiate. And they can be patterned into particular patterns, or you can let them self-pattern via developmental biology. And an example we particularly like is this brain where we have astrocytes, neurons, oligodendrocytes that wrap the neurons with myelin, endothelial cells that provide the blood, and microglia that some of the immune functions in the brain. Here's an example of two of those cell types where the oligodendrocytes wrap the axons of the neurons exactly the right number times the same number you would see in vivo in your brain going white matter and spinal column. And this can be used for studying and even doing therapy on demyelinating diseases like multiple sclerosis. So here's an example where we rescued a mouse with demyelinating disease by injecting a mixture of human stem cells that have differentiated into endothelial cells, oligodendrocytes, and neurons. And so this restores the brain function. So I just want to end on reflecting, putting this in a bigger perspective, on reflecting on alternatives to CRISPR and other previous editing methods. We have rare genetic diseases that are often fixed by adding genes. So that's conventional gene therapy, and that's what this list is up here. Almost all of the gene therapies are adding genes, not subtracting, which is what CRISPR tends to do. Most of the CRISPR therapies are focused on subtracting or knocking out genes. We have, but these have in common that not only are they adding genes, but they're all very expensive, as are orphan drugs in general. These are proteins and nucleic acids that are aimed at a very typically rare disease. We want to bring down the cost, just as we brought down the cost of reading DNA by 60 million fold and synthesizing DNA. We want to make these rare genetic diseases. And one way to do this is preventative, via matchmaking, basically software and genetic counseling. Similarly, with infectious diseases, a great deal can be done with prevention. It doesn't require rare disease gene therapy. And we can monitor the world with a bioweather map. Also, both of those are heavily dependent upon next generation DNA sequencing or reading of DNA. We can increase the success rate of clinical trials by using machine learning and libraries. That's another story that I didn't have time to talk about. And finally, I touched on age-affected diseases. So many common diseases, or so-called complex diseases, are age-related. Even falling down and not getting up is age-related. Almost every disease, COVID-19, has an age component. So we want to aim at those either by the addition of genes, as we just discussed, or possibly highly efficient delivery to many cells. I've mentioned many of my lab members along the way. I want to just mention them again. I mentioned them at the beginning and the end. This is those that are in lab typically wear masks in lab, and the computer people and administrators at home were also wearing masks for this particular lab meeting. So, period, full stop. Thank you very much.
So we have heard a fascinating talk by Professor George Church, highlighting the future applications of CRISPR technologies and beyond. Thanks a lot for him. Now we will move on to the second part of this session, which is a panel discussion by eminent scientists who are panelists during this session.
So I am working as a scientist at CSIR Institute of Genomics and Integrative Biology. My main field of work actually is towards developing or improving the currently existing genome editing systems through protein engineering, as well as through different types of modifications with the CRISPR system. It's not just that we work alone on this, but with a large number of other international collaborators. And we are currently working towards trying to show relatively good proof of concept data for genome editing of the sickle cell anemia mutation in Indian patient derived stem cells. This is something that has been started in 2017, and we are kind of moving towards the second phase of this to one. So that is the main area that I work with. Of course, an offshoot of that is diagnostics that we have recently started, delving into. We have been entering into the diagnostics portfolio a bit, and kind of expanding a little bit our interest in that area, with trying to bring in low cost, more point of care, paper strip based and other fluorescence based diagnostics that relies on CRISPR, and branching out from there in different other ways. But the main focus still remains on gene editing for therapeutic purposes, their delivery and their safe editing case of diseases. Besides this, I also have at least a couple of more projects which involve genome editing of organoids, basically brain organoids, which are mini brains that are grown, that we are using to model neuroencephalopathies and as well as different neural diseases. And we are trying to push in genome editing as a means by which one can not only model diseases but also give us very, very basic understanding of how the disease pathophysiology functions, how the transcriptome gets regulated in such kind of models, and taking on from there, addressing very basic questions of stem cell pluripotency and lineage commitment.
Hello everyone, I am Siddharth Tiwari. I am working as a scientist in National Agri-Food Biotechnology Institute, Mohali, that is under the Department of Biotechnology. And my area of specialization in metabolic engineering of plants, my focus is on wheat and banana. And we are doing work on the enhancement of beta carotene, that is a precursor of vitamin A. And we are trying to increase that beta carotene content by using CRISPR approach, as well as some transgenic or cisgenic approach. Recently we have demonstrated that CRISPR approach for enhancement of beta carotene by the modification of metabolic pathway, that carotenoid biosynthesis metabolic pathway in banana. And the work recently has been published in metabolic engineering. So I am continuing on this direction and trying to develop that edited lines of banana for commercial release, if that guidelines will permit to us. Thank you.
We were an academic institute. We were mostly working in an academic project, trying to identify a way by which we could figure out how to detect the sickle cell anemia mutation on the field, basically. And that's when we thought about repurposing this whole process for COVID-19. Of course, the Sherlock, Detector, Holmes, Inspector, and whatnot, Hudson. There are at least 20 other CRISPR technologies which are already some of them at different levels of development, which has been they're trying to try, and it's getting better. What is really the point to make something which is a detection system which is diagnostically usable? The two main things are, one is that it has to be extremely robust, that it will not be working only in the lab but actually working in the hands of the consumer at every single time. And for that, both the level of reagents which one has to use, and also the entire pipeline has to be very robust, so such that there is a high level of reproducibility. And this is probably the best learning experience for us during the entire 6-7 month journey that we had. Is that it's always good to have to get something working on the lab level. When you can do that, one can have academic papers. But a pandemic like this actually gives you just that right kick in the back, that you need to push it to one level higher, because if you want to take it to the market, then that level of reproducibility needs to be there, which was great. And it is not just a single person, single institute, but almost the entire CSIR and the entire machinery coming together. Finally, of course, we kind of collaborated. We licensed it to Tata, so they also put in all their efforts into it. But it doesn't end there. I think the first version, which has been launched, it's also hitting Delhi this week, and getting into all the hospitals. We are very close to doing something which is can be more point of care, almost at the level of the doctor's dispensary or at the home. We had the first FDA licensed home test approved yesterday, I think 2 days back, probably. So the idea is that testing has to be ramped up for sure, and this ramping up needs to happen at a more point of care locations, where the complexities of sample handling, sample collection can be eliminated slowly. So that is where I think there are a lot of dedicated people are working night and day to get that across, and we'll see how far we can take that.
Thanks, Deblika, for that excellent work from sort of to uh, the application that is very useful for the society in the pandemic times. Dr. Siddharth, you have done excellent work in the agri kind of area where you have engineered the crops such as banana and other things. What is your perspective in terms of the potential in the agri space?
This CRISPR technology has completely changed the scenario of Indian biotechnology or world biotechnology, especially agri biotechnology. That we have noticed that transgenics has certain issues, like there are strict guidelines about that releasing of that, and there are some hypothetical question about that. And most of the countries are not allowing those transgenics for commercial release. But CRISPR has that potential because it's a precise breeding kind of things, you can say. It's a precise breeding. Like that mutation breeding, you can do mutations in that whole genome. There you can get some non-target mutations also. But in case of CRISPR, you can precisely target some of the gene that you want, and accordingly you can generate the traits. So that CRISPR has a high potential, and we have seen that US has allowed so many crops for that commercial approval. India trying to develop that guidelines, and we are hoping that it will also allow us to go ahead with this technology.
Okay. Yeah, that's a great potential which will have a lot of socio-economic impact. So, Dr. Anil, from the industrial perspective, how do you think the CRISPR is going on? Particularly, there will be other issues involved in terms of IP protection, etc. How do you that perspective?
Industry is also eagerly waiting to see how this CRISPR thing takes shape, and a lot of potential. And already Bloomberg predicting that almost 7 billion market globally, from government, from private sector, huge amount of investment has been done. Many startups came up. Big companies are also making different products. So just similar to pharmaceuticals and other industries, we are also pretty hopeful that our government will also, I am sure they are discussing and they will come up with a very positive view of this technology. And I sincerely hope that. And many other countries, especially in the synthetic biology area, they are really moving ahead with this technology and developing many new products, many new commodities, which still not available in India, especially precursor of different chemicals, which could be used for various purposes, from starting from cosmetic industry to petroleum industry, oil and gas and other industries, etc. So I believe, along with other technologies, this will also be something to look forward and hope for in the future.
One thing is definitely the CRISPR and gene editing technologies have a lot of applications. But there are also potential challenges in the area in terms of the biosecurity, or in terms of, making people aware of and adopt and accept, both government and people, the products that are based on genetic modifications. So, Jyoti, what is your perspective? How do you think that we can make these products acceptable to public as well as government?
So that's a valid question, and it's also a valid how to say concern. But it has to go hand in hand with innovation and technology development, because it should not be a roadblock to advancement. I think we already heard from Siddharth, and he's doing amazing work in the field of agri-genome engineering. A lot of times what happens is that due to lesser amount of public dialogue and a gap or a lacunae in the communication with the regulatory authorities, a lot of information, the pros and cons, as well as the actual what are we editing over here, is lost, which is unfortunate, because there is a misconception and a lack of clarity between what is genome editing versus what is let's say gene modified organisms, and due to which there is let's say unfounded fear in certain cases. So that doesn't, of course, say that genome editing is completely safe and it is completely unregulated, but it also tells us that there has to be regulations which permit both the development as well as the mitigation of unwarranted fears through communication and dialogue, and at the level where the policymakers can actually take into account.
Thank you for that perspective. Dr. Siddharth, I think it is even more sort of applicable when it comes to agri or food kind of areas. How do you think we should be going ahead in that direction?
I think it's a high potential, as Devojyoti already told about this technology. Although it has to be regulated at some level, it should not be let like that a completely free, anybody can use as they want. There should be some regulations, but it should not be strict like that transgenic, because everybody knows that it's not like that kind of things as people noticed about transgenic, because you are modifying the genome, that gene in the genome itself. It's, you are not taking another gene from other place and introducing in the genome. That is I think a government is also trying to relax some kind of things, specifically like SDN1 or SDN2 kind of mutation.
Okay. Okay. Thank you very much. So, Jyoti, another interesting thing will be that there is always a reservation from the academic perspectives in terms of developing technologies that may be marketable. Your lab has really achieved that. So what are the challenges you see in terms of academic scientists developing tools and technologies, particularly in the CRISPR area for markets?
So, so I think it is something that is kind of an academic industrial ecosystem, which is a little bit, let's say, we are catching up. I won't say it is not there, but we have a lot to do. We have a lot of catching up to do, and this ecosystem actually allows academicians to work closely with industrial partners, whether through startups, collaborations, whatever mode it might be, licensing and so on. Somehow, we do have delays which normally happen a little bit at the level of bureaucracy sometimes. But all in all, even though the ecosystem is there, we have not been able to tap into it to our fullest extent. The pandemic was a very, very eye opener, because it required things to move at a fast pace, and because things had to move at a fast pace, a lot of things which would otherwise take years to do were kind of made rapid. And that is how it ideally should be. Innovations and technologies should be move at a pace where its requirement should dictate how fast it should come to the. So I think that the pandemic has probably highlighted. And I mean, I would never say that we do not have an ecosystem which promotes it. I think an ecosystem, but unfortunately we do not realize the full benefits of it due to delays at different levels, which is getting better, and which we can definitely learn from the pandemic situation.
Okay. So, Dr. Siddharth, as Professor George Church was also sort of mentioning in his talk, of course, the revolution started with CRISPR technologies, but there is a lot more happening beyond, and certainly there are other areas which are developing, and technology is also developing. How quick are we in terms of adapting, going beyond CRISPR, in your opinion?
Yeah, I think every technology has their own advantages as well as disadvantages. We have to see that advantages, like transgenic people are saying it has some issues like that. But I think so. We have very specific guidelines, regulatory guidelines, and biosafety guidelines. And if people are following those guidelines, we have to be allowed those technology also for commercial use. We should not be like that. If other countries or other people are saying there are some issues, it should not like that. If that scientists are following guidelines and they are proving their technology, it is workable. And so that I think government should think about this approach also. This is my personal opinion. Yeah.
So, Dr. Debjyoti, what do you think about the technology which is really moving very fast, and how are we adapting to that?
So I think we have very little time, from what I understand from the host writing there, but I just in a single sentence mention that we need to move from catching up to actually being part of the technology, which is unfortunately true for a large number of innovations. And this is moving at such a fast pace, like Anandu had mentioned right in the beginning. Almost daily or a weekly we have new innovations coming in. So we need to really move from a point where we are catching up and taking something, adapting something in our own ecosystem, to really encouraging, promoting, and funding science, which is asking very ambitious questions. Unless we do that, we will not be able to be a part of a technology and develop a technology. We'll still be doing copying, and hence I think that is where the impetus should be.
Right. I think the time is up for us. First of all, thank you very much for participating and giving your time for this discussion. We really had very different perspectives from industry to different areas of technologies, from agriculture as well as in the biotech or diagnostic space. Thanks again for each one of you, Anindya, Siddharth, and Debjyoti, for participating. Thank you very much.
Thank you. It's a pleasure.
Thank you so much. Thank you so much. It's a nice meeting.